Okay, so I'm just going to talk a little bit about finding SNPs in transcriptome. I'm going to show you a combination of some background slides that I have in my other classes, the project we did a long time ago with specific herring in a transcriptome, and then that was done with CLC. And then after that, I'll show you how you could do it with free software. But if you just conceptually think about it, I'll just act it out. I mean, I guess I could talk about it. You know what I mean, so these are just the educational slides, just look at population genetic variation. It's important to know there's differences in species, okay? In a transcriptome, not necessarily, you have a wealth of information beyond gene expression, and we really haven't talked about measuring expression levels, or if they're there or not, but you could also mine it as SNPs. It's not, there's caveats because there's no eukaryotes, you don't have, you've got introns and exons. But if you've got a bunch of a million reads lined up onto a gene, you've got the ability to determine if it's an all T or in one population or all A and all G and all C. But you can actually find SNPs in transcriptome data. And all really you have to do is something as simple as if you have the alignment, and then you're just looking for at every single solitary base, is it homozygous or heterozygous? So is it AAA or AAA and half OMT and so on. So imagine a computer can do that. I mean, my interest, as I've talked about before, is more from a physiological perspective, so I've always been more interested in kind of a SNP, a change in a nucleotide that would result in a change in a protein that you would think might have a different function. That was years ago. Now we know that, with this epigenetics, that a SNP that might not necessarily be a gene could have just as much or even more of a functional effect. Okay, but I got into it because I was really interested in the genes themselves. And just giving you again, this is what I teach in this graduate classes. You can imagine if you change one of these nucleotides, there's a possibility you're going to change an amino acid, which changes a protein, which could change function. Let's see what we have. There's a quiz, but you get the idea. So real quickly, so CLC is software that costs money, that you pay for. It's not open source, but we do use it. This is probably an older version of it, but you have a directory structure. You can import data, you can do assemblies, and you can find SNPs. So this was a project several years ago. This just kind of walks you through. You would do the assembly. So you could also use Trinity, the assembly. You could we actually use Top Hat, which I'm going to show you in a second. Top Hat for finding SNPs now, but it's analogous in this particular software. The assembly is what stacks all the reads together, so you get this. Okay. What it looks like every step. It gives you an example. So if you wanted to look at a specific loci to see if it was heterozygous or homozygous for a specific nucleotide, you could picture it. The talk I gave along. Same slide, huh? Lazy. So we had 4 populations of herring just walk you through it. This is like a history of it. What is this? Does anybody recognize that machine? 454. So we used that. What, does anybody recognize that machine? Solid, yeah. So for this particular project, we did a reference with 454, then went back and did short read solid to do transcriptome with a lot of individuals, gene discovery, looking for SNPs, and then partly genotyping. Again, this is kind of outdated, but it's fun to look at old stuff. Gives you an idea of kind of what our output was years ago in terms of coverage and consensus length from the 454. Another way to visualize the coverage, so that's the depth, which is important with up there. This is something that you could do, that we could definitely do in class this week with open source software to create heat maps. This one just is between what? Huh? Liver and testes, yeah. So we did we happened to do 2 tissues. That's all that. So this is just features was a way of saying genes, so very analogous to what you could look at. This is another great example of if you wanted to. We talked about these terms, but this is one way to take a whole transcriptome. Remember, and then we annotated it with BLAST, and then we connected it with gene ontology. And then there's these very high level, gene ontology is a hierarchical system, and at the very top, we call them go-slim terms. And they're in 3 different kind of tiers, right? You've got cellular component, molecular function, and biological process, yeah. These are the biological process slim terms. So if you look at the whole transcriptome, most of it has to do with RNA metabolism and protein metabolism, and you can, there's very few that are involved in, developmental or death or cell organization. But it might be that you are an expert in cell adhesion, right? And you wanted to see what these genes were. You imagine there's a table somewhere, you could grep all those, and then you could focus in on that. So this is one way to represent describing the function of an entire transcriptome. And this is just a comparison of like an enrichment. So if you look at, these are where the SNPs were, and that's where all the features of the one that I showed you before. So if one piece of the pie was bigger, that would have been more enriched for SNPs. So you can imagine, you can see, are SNPs more likely to be in one biological process as opposed to the other? You think, if you were just, without looking at data, would you think that's possible? Yeah. I mean, if you just think about it, if you put a SNP in a very important gene, you might die. If you put there might be some value of having genetic variation in ones that are involved with, responding to a pathogen or something. But it gives you good ideas, a way of. These are just, nice. This was a phase where I really liked pie charts. Again, so this is a nice example of, we were really interested in looking for SNPs in the stress response, because we were trying to see if I didn't tell you the whole backstory, but the Exxon Valdez, the crash, oil spill, all the salmon herring died. They didn't know if they were coming back, and if there was population structure, and could there be population structure driven by the response to the oil. So we wanted to really look with the SNPs within just genes involved in the stress response. That was one part of the pie before. With the gene ontology, we could either break it down to the subcategories, all of these different processes, if we wanted to narrow it down. Okay? And this should look familiar. These 6-digit codes, these accession numbers, and I probably did blasted and joined it and got the name of the gene. And so some of these genes, if you're into the environmental stuff, aryl hydrocarbon receptor, superoxide dismutase, these are involved in detoxification processes. We found some SNPs in there, and so we wanted to kind of keep working on it. What was that? This is, I was very proud of myself years ago. So we'd do everything on we had to do everything ourselves. No really value in this, but we did this is how I had to come up with a way to determine whether a SNP was synonymous or non-synonymous, so whether it affected the amino acid or not. When all I had was a nucleotide sequence. There's no way I can remember what I did, but I take these sequences, and you can run it through a program, get ORF. What do you think get ORF does? Gets the open reading frame, okay? So that just gets that. So that just really just looks for start and stop codons. I would have done this in the command line, just like you did BLAST. It would have probably been, get ORF in out. Okay, it's very similar. I assembled those. Look for SNPs. So stack them, find the SNPs, and then annotate after the fact, and then filter based on E values. Okay, so we've made like fake proteins based on open reading frame. Then if it blasted as something, I would have been more confident that it was really a real protein. I'm just hoping, and that's what I mean by filtering on E values. Okay, so it had to have a big open reading frame. It had to have a very strict E value, and then it was likely to be non-synonymous. There's different ways to do it now. But hopefully this should make sense and conceptually of how when I went through the pipeline. And 7,000 SNPs were discovered, 25.14 were non-synonymous, and I really got crazy with my pie charts. Right? So this is breaking it down. So it was the non-synonymous ones that changed the amino acid. And then, of course, so there's a theme here. Lots of data, you subdivide it, and then you make a pie chart of it. So these are the breaking down now, just looking at the non-synonymous, what categories they fit in. That was a comparison, which we'll get to either tomorrow. Tomorrow's Friday? Yeah. Or Saturday. Kind of conceptually doing a better enrichment analysis. This is just pictures, like which ones are more enriched for our processes. I'll show you how to do that statistically and get values. Again, breaking down just the non-synonymous genes with non-synonymous SNPs into the really stress. Yeah, exactly. Yeah, so all that's published, and we did all kinds of fun stuff to see. This is early on we were trying to decide what do you call these curves, but this is how you can evaluate if you keep making new discoveries by sequencing. What's this curve?
Saturation.
Yeah, saturation level. This is what we were trying to assess between the two libraries. Where every time you went up more reads you sequence, you would get more context, right? So if this flattened out, and you still got 30,000 contexts, 30,000 contexts, 30,000 contexts, and 30,000 contexts, then you really didn't need to spend all this money sequencing. That's kind of the assessment we made on our sequencing effort. And these is just looking at, then the actual population, the FST values based on the SNPs, and the different population, but that gets into more of the biology. Brent, what is that?
That's just a scatter plot.
FBT. And the different colors are the different populations, and those I'd have to look at the code, but those refer to the gene that those SNPs were found in. Again, this is all published, so I can post it. You can look at the whole paper, and you really like it. I don't know if all the pie charts are in the paper, but and so this is old too. This is a separate thing. So if you're doing genomic data, RADSeq, that type of stuff, you would use, there's special programs for that. So what I was showing you is short read sequences for whole genes lining up and find SNPs. You've got things such as stacks. That's kind of the popular open source thing for looking at RAD data, where you just get a bunch of genomic data. The same idea. It stacks them up and it looks for SNPs and does finds the variance. Yeah, for RADSeq exclusively. I mean, that's what it's made for. You wouldn't even use it for the gene stuff. This is just part of what's inside. So when we do RADSeq stuff, we use stacks. So that's, again, stacks. You could do it. It's just like BLAST. It's a couple more programs, but U stacks, C stacks, input, output, a couple variables, and follow the instructions. So the same kind of interface. But real quickly too, so how would we do this now? The first thing I would do is, RNA-seq data. I would do this. Get an assembly, okay? And the common assembly stacks, I should use that word, the common kind of alignment of a bunch of reads is a SAM format, okay? S-A-M, that's the format, it's a generic format. Top Hat is made for doing that for RNA-seq analysis, but it creates a SAM file. So Top Hat, just another program like BLAST, like Top Hat input output. Okay. We'll show you again. I happen to. It's kind of resource intensive, so we again use iPlant for this sometimes. Okay, to do Top Hat. So this is the iPlant environment when you log in. You've got data, apps, analyses. So when I do, you want to run an app, you could click on this box. And this, if you look on the side here, they have organized them into categories. All right, NGS, next-gen sequencing, transcriptome analyses, tons of things. If I just wanted to see, you could also just search for an app. So this is all the Top Hat versions they have there. What iPlant has done is it's taken all of these programs you run at the command line and made a somewhat simple interface. Okay. So that instead of typing -in file location, click the button, show me where your file is in the cloud. Okay. So, for example, if I want to do a Top Hat, I would pick this Top Hat. It's version 2, so it's after version 1. I pick PE because that just happened to stand for paired in. And then it goes through, and if you want to add your file, you click at the Add button. It brings up a window. Your data's there. You navigate to click on. Okay, so you could go through that kind of. You fill out all that stuff. Hit submit. It submits the job to the cloud, and it runs and runs and runs. And if you haven't said, it'll email you when it's done if there's not a failure. Okay. When you do that in Top Hat, your output's a SAM file, okay? And then what you can do with the SAM file is there's a whole suite of tools for working with SAM files called SAM tools, yes. and so it manipulates those alignments. It can do a lot of different things. MPileUp is the one that actually gets it for calling the SNPs. Okay. You convert the BAM to SAM, so you would input it and you would run it. So I've already done this, so I'll just show you what it looks like. So the other thing, once you run those jobs and they're finished, they show up here. This is your analyses. Okay? And I've got a lot. You could see Trinity and a bunch of other stuff going on there. So I just searched, and these are results. Okay, so the results are just spit out in a file. So the value in, the reason I would ever use something like this if it was something that took a long time, right, and I didn't have the computer resources, or if I wanted to multitask and run it locally and run it in the cloud. And it spits out a VCF file, okay, and then you can do different analyses on that. You can actually visualize this in a genome browser too, and you could download it. Okay, so this is the actual just text file, but you should see something that's somewhat meaningful. These codes for the genes, I have to look. This is the position. This is the what it is in the reference, what it is in terms of the SNP. Okay. So I've showed you a couple of things there, how to do it open source, and also showing you again roughly iPlan as a tool you could use. But again, I just want to remind you that SAM tool, you could do all of this on a computer, maybe not the one you're sitting in front of now, depending on the amount of data you had. But you can go to download the code. Actually, a lot of places use GitHub for what it's meant for, the actual storing software. You could download SAM tools, and a lot of great tools that are community driven like this, and bed tools, which I'll talk about tomorrow or the next day. What do you think bed tools is used for? For bed files, right? But this gives you an idea. Again, you should feel so confident. You understand what all these things are. You're running a program. You're putting some flags. You're probably telling it where the file is. I know it's small, but what is that? A pipe, right? And it's then it's running another program, flags, pipe, and it's redirecting, and there's your output. Okay. So you should have a little bit of confidence now that if you wanted to explore a new program, you could at least understand the language. And if you get a page like this, they do a great job of explaining, every column, what the output is, what it means, and so on. All right, so that was just a quick tangent on how you could look at transcriptome data, mine it for SNPs, do whatever you want to with them, whether population genetics, and the open source tools that you could use to interrogate the data that you have or will have. All right. So I'll be quiet for a little bit. You can work or play. Orange sticker if you need help, and then I'm going to pick back up on the gene project and where I left off of downloading my FASTA file and what I would do next. Okay?